thank you all for being here. And we continue our seminar series, which we started a week ago, with a friend who came here all the way from California, Professor Fernando Santana. Professor Santana is originally from Puerto Rico, where he got his undergraduate degree. He then went to the University of Maryland, correct me if I forget, went to the University of Maryland where he did his Ph.D. and continued to do a postdoc with John Lever. And then after that, I think Vermont, with Nelson, Mark Nelson. So that's training-wise, and then he spent many years at the University of Washington in Seattle, and only recently, as of July, he moved to University of California at Davis and assumed the position of the chair of Physiology and Membrane Biology department, and a few other responsibilities. So there's mentoring people and so on. So Fernando is really well known for his very creative work and creative approaches to experiments. And he's done beautiful experiments of calcium cycling in cells, mostly cardiac and smooth muscle, where he combined calcium and electrophysiology to study mechanisms of arrhymnias. Recently, he's been very interested in arrhythmias in the context of heart failure. And so, Fernando, thank you for coming.
Thank you, thanks for the introduction. It's always a pleasure to be here. Well, one thing that I will tell you, as you can tell, I have an accent. So if I say something that you don't understand, or formulation, the grammar is not correct, feel free to stop me and I'll repeat it. And related to that, I want to warn you that if you have your iPhone active, Siri can hear me. And I was in this meeting, Dean was there, and as I was giving my talk, somebody had their iPhone on, and Siri says, "Could you please repeat that? I couldn't understand." So of course I had to repeat it. And it's not the first time. I have a Korean car. It's a great car. It's a Kia, and I tried to use voice command, and the car just simply cannot understand what I'm saying. So no problem. But remember that as far as I'm concerned, you're the ones with the accent, not me. So if you guys with your strong American accent say something that I don't understand, I'll make sure that I'll ask.
It's all right, it's okay. My car asks me, "Are you from Israel?"
So as Yoram told you, my lab is very interested in the mechanisms of calcium dynamics and smooth muscle and also cardiac muscle. And something that an issue that came to the forefront in terms of our research about 4 or 5 years ago was in fact brought up by a student in one of my physiology lectures to medical students. It was this issue about how come the reproducibility of a contraction beat after beat after beat is so high. The coefficient of variation of contraction in the calcium transient, one action potential after the other is very low. It's amazing how reproducible it is. And he asked me what were the mechanisms underlying that, and really that triggered a set of discussions that in many ways took us to some of the stuff that I will be presenting here today. So just as a matter of giving you that introduction, let me take you through the cardiac cycle. So everything begins, as you know, in a specialized set of cells in this yellow ellipses there that have the ability to fire action potentials from time to time. And here's just a set of action potentials recorded from inducible pluripotent stem cells that have the nodal phenotype, just to give you a sense of what that looks like. These SA node cells fire these action potentials, as I said, in a continuous manner, about 1 per second in a human. And that this action potential travels from one cell to the other via gap junctions, as you know. First, depolarizing the atria, producing action potentials that are relatively short when compared to the ventricular action potential. that is not triggered until after a delay, because all the electrical signals on the physiological conditions that will electrically activate the ventricles has to go first to the ventricular node, which also has the ability to acquire action potential spontaneously, but normally doesn't do so because it's taken over by the faster pacemaking activity of SA node cells. So that's the cardiac cycle. In the system, the signal for contraction is obviously that electrical signal. And this one thing that keep in mind is that this cycle will be repeated 3 to 4 million times within the lifetime of the average human. So that's a lot, okay? And a concept that neuroscientists lead with, but that is completely foreign to cardiac physiology is the concept of failure. And by that what I mean is that every time an action potential arrives to a ventricular myocyte or an atrial myocyte, there will be a contraction. It doesn't really, in some cases it will be weaker, in some cases it will be stronger depending on the physiological conditions or the pathological conditions. But there's always going to be a calcium transient that will then trigger contraction. Always. That doesn't happen obviously in neuronal communication, but here that is the case. So that is of the essence, what has been driving our research over the last 5 years. The potentials of the SA node cells are relatively complex, particularly when you compare it to action potentials of skeletal muscle fiber, which are very short, about a few milliseconds long, or a neuron also a few milliseconds long. They go all the way down to maximum diastolic potential of -65, and then spontaneously depolarize until they reach a threshold, and then there's a relatively faster rate of inactivation. The tonic current, which is produced by HCN2 and also HCN4 channels in SA node cells, but also the calcium clock, as Ed Lakata would like us to believe, to be playing a role in the depolarization, spontaneous depolarization of these cells. Then there are two sets of voltage-gated channels that I want to focus on. Number one is one called CAV 1.3, and these are the hydropyridine-sensitive voltage-gated calcium channels that are very similar to CAV 1.2 channels, also the hydropyridine-sensitive channels. And CAV 1.2 are the predominant or the only voltage-gated calcium channel that is expressed in ventricular and in myocytes. So when a ventricular myocyte receives an action potential, the calcium channel that will open in response to that depolarization will be CAV 1.2. But SA node cells express CAV 1.3 and CAV 1.2. The difference between the two is that CAV 1.3 channels will open at a much more negative membrane potential, and hence this current that is driving the membrane potential of this section here in the early onset of the action potential of SA node cells. Once these two channels inactivate through voltage-dependent mechanisms and also calcium-induced inactivation of the channel, then the gate current voltage-gated activated channels that take over, bringing the cells back to the diastolic potential, and then having the cycle start all over again. But I will tell you the story that I will tell you today, primarily concentrating on CAV 1.3 channels of these. Now, CAV 1.3 channels are expressed throughout the nervous system. So many of the things that I will be discussing here not only apply to how we think about SA node cells, but I also want you to think about hippocampal neuron cells and other neurons that express CAV 1.3 channels. And I'll show some data on these effect cells in experiments that we did recently in the laboratory. So there are two isoforms of the channel generated by alternative splicing of CAV 1.3. First, it's one so-called CAV 1.3S, for short. And the main difference here, aspect of this channel, is that it has a relatively short C-tail when compared to the long form of the channel. It has a very extensive C-tail, about 3 times the length of the CAV 1.3S channel, with two important regulatory domains. One is the proximal C-tail regulatory domain, which is shown here in gray in this cartoon, and the distal C-tail regulatory domain that is expressed in this almost at the very end of the C-tail of the channel. What I'm showing you in purple is the IQ domain, and in green is the pre-IQ domains, and these domains will become important, and also this distal C-tail regulatory domain towards the end of my talk. But the general consensus is that in the C-tail, of CAD 1.3 S channels. Calcium binding to the IQ and pre-IQ domain is what dictates how long this channel opens, primarily by controlling calcium-dependent inactivation of the channel. Situation is slightly different in the long form of the channel because the distal CTL regulatory domain, the DCRD, will then interact with the IQ domain and hamper and prevent strong interactions between calmodulin and that CTL. But if we think it has additional, in the distortion of the CTL, but we think it has additional calcium costs. So remember, two isoform of the channel, the long and the short. They have distinct pharmacologic, biophysical profiles. Here's a set of currents recorded in the presence of barium in extracellular solution, depolarizing from -70 to -10. See a nice inward current that inactivates very little over time. There's a little bit more inactivation in the short form compared to the long form, presumably due to voltage-dependent inactivation of the channel. And by the way, these recordings were done in TSA cells expressing either the long form or the short form of the channel.
With no accessories, nothing?
Yes,
No.
there's a full set of accessories within it, and I'll tell you in a second. So once we switch the solution from a barium solution to a calcium solution, you see here that then there is a distinct inactivation of the channel, of the current. The rate of inactivation seems to be faster in the short form compared to the long form. But the thing that struck us was the fact that switch going from barium to calcium in the short form here didn't translate into a change in the amplitude of the current in the short form. But look at what happens in terms of the amplitude of the wholesale current in the long form. There was almost a decrease by about 50% in the amplitude of the current. So that told us that maybe something was different in terms of potentially either the permeation of calcium to the channel or the way that the channel is controlled by the calcium that flows through that channel. Here I'm showing you just a set of current voltage relationships from the currents like the families of currents like the ones that I showed you in the previous slide in the presence of barium and then calcium. And then for every experiment we would go barium, switch to calcium, and then run a full IV in each one of these, in a solution containing either one of these bivalents. And again, just to highlight the point that in this long form, the current decreases by about 40% in the presence of calcium, but not in the short form. They're about the same. So what's going on here? So first thing that we did was to try to understand this mechanism, and something was that, well, maybe the single channel current, the amplitude of the elementary currents produced by CAD 1.3 channels in the presence of calcium, and the CAD 1.3 long isoform in the presence of calcium is different. That's not the case. In these recordings, this is at -30, depolarizing from -80 millivolts. We could see distinct openings. There's 20 millimolar calcium in the patch pipette solution. These are cell-attached patches. We see distinct openings of what appears to be a single channel at this level, about 0.5 picoamps in terms of amplitude, with occasional openings, simultaneous openings of two channels. Now, one thing to notice is that not only the amplitude of the elementary event, which we think is 0.5, as I said, was similar between the long form and the short form, but also we saw many frequent multi-channel openings and closings in the short form of the channel. And these were very rare, if ever saw, in many experiments in the long form. Okay? So the same single channel conductors, and yet they seem to have more multi-channel openings. What's going on here? Keep that in mind. We'll return to that concept shortly. We're confident that what we're recording are CAD 1.3 currents of channels, because when we do generate ensemble currents, we can see nice activation, and then just by averaging many of these records like the ones that are shown at the top, and inactivate as a function of time, likely due to calcium-dependent inactivation and also voltage-dependent inactivation of both the long form and the short form of the channel. So the relationship between the single channel current and the wholesale current, macroscopic current, is very simple. simply mathematically, from a mathematical point of view. The whole cell current equals the amplitude of this elementary current times n, which is the number of channels that you have that are functional, and the open probability of these channels, that particular patch or cell membranes, okay? So we know now that the elementary currents between the long form and the short form are about the same, okay? About 0.5. So something probably is happening in terms of MPO for these channels. What we did was that knowing the whole cell current and the amplitude of the elementary current at that very same potential under the very same ionic conditions, we went back and calculated the MPO of these channels. Now, we're changing the solution over a matter of minutes, so we don't think there's any change in the number of channels in the membrane. So we're going to assume now that the MPO value here, we can get, really forget about no change in N. So therefore, the change that we're going to see is most likely due or dominated by changes in PO. When we came up with this calculation, we saw that in the presence of barium and calcium, there was almost no change in PO from in each one of these channels. So this bar here connects what happened during each one of these experiments. But look what happened when we did the same analysis now with the short form of the channel, CB 1.3S. It was almost in every case we saw an increase in NPO, which makes sense. If there's no change in the amplitude of the single elementary current that is being produced at a particular voltage, therefore the only thing that could be explaining the fact that the current was different was that there was an increase in NPO. That, was summarized in this scatter plot here. What we found was that there was about 1.25-fold increase in NPO, higher NPO, in CAV 1.3 short channels compared to long channels in the presence of calcium. So these two isoforms seem to be differentially regulated by calcium flowing in particular through this channel, and that can take place when memory depolarization. So clearly, there's something going on different, potentially, obviously, concentrated at the C-tail, but we wanted to figure out what exactly the mechanisms were, the molecular mechanism. Now, recording single-channel activity from TSA cell expressing CAV 1.3 channels, or even an SA node cell, is technically challenging. And one thing is that landing a patch pipette into a region of cell where you have a channel, or a single channel, or a cluster of channels that can work in unison could be quite difficult. It's very good in the sense that you have very high resolution recordings of a single molecule, in a particular case, or small cluster of channels. But if we wanted to sample a larger portion of the membrane, what we can do is to use an alternative approach. And the fact that these channels allow calcium into the cell, and that we have really nice calcium sensors available to us, and very nice cameras and lenses and microscopes that will allow us to record these calcium influx events at a high level of resolution, prompted us to see whether we can record these calcium signals resulting from single or small clusters of CAV 1.3 channels, and see if we see any difference between the long form and the short form that will be consistent with the electrophysiological experiments that I showed you before. Here's just a movie of a calcium sparklet, one of these TSA cells. There are a couple of events, just one here. Maybe showing another one around there. These are the calcium sparklets right there. We do not see events like this when the cells are not expressing these channels, or we express other types of channels that don't conduct calcium. So we're confident that these are calcium events being produced by the opening of voltage-gated CAV 1.3 channel. The thing that we saw that was interesting was that we can find locations of the cell in which, like this one here, which show repetitive opening of voltage-gated calcium channels, producing what appears to be quantal unit of influx of about 40 nanomolar, which we think is the result of the opening of a single CAV 1.3 channel, based on other experiments that I'm not going to mention now. And occasionally, like what we saw at the single channel, with the single channel recordings using the cell attach mode of the patch clamp technique, we would see what could be the result of a calcium influx due to the opening of two CAV 1.3 channels opening simultaneously. This is all with the long form of the channel. But look what happened with the long form of the channel. Now we were able to see many events at the quantal level, about 40 nanomolar, which is expected given the fact that the single unitary elementary currents that are produced by the CV1.3S and L is about the same, show you the previous slides, but now we saw many more events resulting from the opening of 2 and up to 3 channels, opening and closing presumably simultaneously. These 2 values that I'm showing you here, this kappa value here of 0.03 and this kappa value of 0.029, these are coupling coefficients, meaning by that the probability that the 2 channels or the cluster of channels will be opening and closing in unison. And a coupling coefficient of 0 amounts to pure stochastic opening of the channels, and a coupling coefficient of 1 means perfect coupling of 2 channels opening and closing all the time. I can tell you that on average, the coupling coefficient of CAV 1.3 long channels, is in the Sparklet recordings, was about 0.05. That was the average. It was very low coupling coefficient, purely stochastic activation, at least on the basis of this analysis. But the kappa value of CAV 1.3S Sparklet sites, like the one that I'm showing you here, came to an average of around 0.25. So it's not a very strong coupling, but there were many of these sites, particularly with this isoform of the channel, that were able to open and close simultaneously. Okay, so maybe these 2 things, as I said, are linked: the couple activation of the CAV 1.3S channel and the increasing NPO open probability that I mentioned in the previous set of experiments. Now, one hypothesis that we had for the CAV 1.3 channels, and that was obviously that was motivated by many experiments that we have done with CAV 1.2 channels, is that possibly, like CAV 1.2 channels, CAV 1.3 channels that are clustered together, that are close enough, can interact and via C-tails. And if that were to happen, remember the polarization would increase the open probability of joint channels. The way we saw it was that, well, maybe what happens is that membrane depolarization activates a CAV 1.3 channel that allows some calcium going through the cell, and that somehow links these the short channels in such a way that then the channels that are touching each other, potentially via the C-tails, can have a higher open probability and potentially a higher coupling coefficient, essentially leading to cooperativity of voltage CAV 1.3 channel gain. Now, if this is the case, the first thing that you need to find out, or we needed to find out, is whether these channels clustered. That's the case. I'm here showing you a set of images collected using a TIRF microscope, a total internal reflection fluorescent microscope. This is just showing you the fluorescence just right, within the evanescent field that is created when you hit the critical angle for total internal reflection. This is just basic fluorescent measurements with some clear clustering around these areas, here 1, 2, and 3. But in this preparation, we use a different, an extra level of, where we achieve an extra level of resolution. We use a ground state depletion system to then try to go from a level of resolution in the order of 250 nanometers laterally to about 100 nanometers in the z-axis, to a level of resolution of about 30 to 35 nanometers. And are you guys familiar with GSD storm imaging? Do you want me to go over it a little bit? Do you? So the way it works is that basically it's based on Heisenberg principle of uncertainty, and how the more information you get about the location of a particular photon, the more confident you are about the exact location of that photon. So with GSD and storm, what you do is that you, first of all, you try to excite very few molecules. In this case, what you do is that you bring a lot of the molecules to the dark state and then slowly bring them in to the ground state and then cycle fluorescently, so that you can record one molecule here, one molecule there, and then you accumulate the locations of these flashes of light. You have to use very high, very sensitive cameras, and then you accumulate that information and you start building up localization maps of these photons. And based on the number of photons, that's what gives you the level of certainty of a particular location. When I talk about that, that I have a level of resolution of about 10 nanometers here, that means that that's my margin of error. That's where more or less the location of that molecule, the center of mass for that molecule. Anyway, you can see that you can break down these smear areas even further and see individual puncta of CAV 1.3 channels. And one thing I will tell you is that they vary in size, ranging all the way with an average size of about 2,500, 2,100, 2,200 square nanometers. But if you look at the histogram distributions, varies quite a bit. So what that suggests to me is that the number of channels that are forming each one of these clusters varies to some degree. Now, going from this type of number to the particular number of channels that are within each one of these clusters is very difficult. We're using a primary antibody and then a secondary antibody, so the fluorophore is just dangling, about 10 to 15 nanometers away from this, where this antibody is binding to the channel. So keep that in mind. So to count the number of channels in any one of these clusters, then you have to take a different approach. And the way we did it was to then express fluorescently tagged CAV 1.3 channels. In this case, these are all CAV 1.3S channels. And we did the same experiments that I will show you with the long channel. And what we found was that there were some sites that if you monitor over time will show a single transition, fluorescent transition like this, which presumably results from the bleaching of a single EGFP, and therefore a single CAV 1.3 channel. That presumably is just one channel in that particular location. Now there was another site within this cell, this site over here, site number 2, that had larger transitions. In this case, had 4, 6 steps. So presumably resulting from a cluster of about 6 channels. In other sites, like this one here, with about 4 channels. We analyzed many of these cells, and I can tell you that on average, what we found was that the number of steps per cluster was about 4 channels. Okay, and that matches fairly well with the sparklet recordings in which I show you that there were events that were all the way to about 3 channels opening and closing in unison. This is some of the areas that we average doing that, and that would correspond if this were one-to-one relationship between an area of 1 to 1.6 a block, a cluster of 1.6 square nanometers, presumably will cover between about 2 to about 4 channels at the maximum. That in fact matches pretty well with what we know about the molecular dimensions of CAV 1.2 channels, based on dry electron microscopy. So yes, these channels tend to cluster, and they are within about 30 to 40 nanometers away from each other. It's in super resolution, and there are about 4 channels in each one of these clusters. And keep in mind, not all EGFPs are fluorescent. So this is a lower end estimate. So at a minimum, you have 4 channels in these clusters, but it could be higher than that. So what we did was that, okay, so they're together, no question about it. And one thing that I will tell you is that the short form and the long form has the ability, both of them have the ability to cluster. So all the things that we saw from using sparklets and also using single channel recording seem to be independent of their ability of the long form and the short form to form clusters. Both of them seem, for whatever reason, to gang up together. Something is controlling this that we don't know yet, but something that these channels have that ability. We use a dimerization system called the cryptochrome system, composed of 2 proteins, CIBN and TRI2. CIBN is light sensitive. You hit it with 480 nanometer light, and it changes its molecular complement. It's the way the molecule creates it's organized that creates a pocket where TRI2 can bind. And the good thing is that this interaction is reversible over time. So what we said was, well, if these channels are close enough and they can interact, then when we hit them with 488 nanometer light, then CIBN and TRI2 can touch each other, and presumably, if that's all it takes to get these channels going, these channels will start opening and closing simultaneously. These experiments obviously imply that we are forcing the interaction between these 2 channels. Okay? It's not physiological means. It just means that, well, if we can force this interaction, what happens? Well, what we found was that expressing CIBN alone, fused channels, CIB 1.3 channels, was sufficient to put functional channels in the membrane. And if we illuminate with 488 nanometer light, we did not see an increase. If anything, in this particular example, there was a small decrease in the amplitude of the current. This was all attributed to normal roll down the current, well-defined phenomenon. But now when we express the CABN form of CAB 1.3 and CAB 1.3, these are all the short forms of the channels in Karate 2. Every time that we illuminated with a 488, we would get an increase in the amplitude of the current. As if now the open the activity of the channels would go up after presumably these two channels are touching each other and fusing. And that increase was about 30% in terms of the peak fold, the fold increase in the peak of the calcium current. Now that's all good, in the sense of, well, if you force the interactions, that's what can happen. But we wanted to go into what would happen spontaneously. Are these channels capable of touching each other in a spontaneous manner? And for this, we use a different approach. We use a bimolecular fluorescence complementation assay. Here you take a YFP, you chop it in two, and when you have the N portion of the YFP and the C portion of the YFP, they're non-fluorescent. But if you bring them close enough and they can reconstitute into one functional YFP, then fluorescence can be achieved. Okay, so you can go from the dark state when these two proteins are apart to a bright state when the two proteins are coming together. And according to the model, what we thought would happen is that as we depolarize, calcium comes in, somehow that brings the C-tails together, and if that's what's happening, and we get a reconstitution of YFP, we would get fluorescence as a result of the channels opening and calcium flowing in. First, what I want to show you is what happens in the PSA cells. These are third images, so you're seeing just the footprint of the cell in the presence of 2 millimolar barium. We depolarize to multiple membrane potentials, all the way from -60, -20, 0, and 20. No change in split venous fluorescence. So these two channels don't seem to be touching each other at all. Okay, this is all with a short form of the channel, by the way. Yeah. But look at what happens when we have 2 millimolar calcium present. Fluorescence progressively increased as the cell was depolarized all the way to positive 20. And this is summarized here over a longer period of the wider range of potentials with the venous F or F naught. Everything was normalized to the basal fluorescence. And in all cases that we examined, in the presence of barium, there was no increase in venous fluorescence, no reconstitution. And in the presence of calcium, there was almost a twofold increase in average fluorescence. So calcium flowing through these channels seemed to be very important in bringing these two channels together, and presumably, causing the coupling behavior that we see at the functional level. One problem with this system that we try to turn in our advantage is the fact that once venous is reconstituted, it will not be reversible. You're stuck with these two channels interacting with one another. So what we did was to depolarize to all the way from -70 at different steps to positive 60 in the presence of 2 millimolar barium or in the presence of 20 millimolar calcium. And here there was no reconstitution, you saw the experiments. But here in the same cell, with calcium, presumably you have reconstitution, you saw it in the previous slide. Now, if these two channels, by the fact that they're touching each other, then can open at a higher, with a higher probability, then you would expect that on return to polarized potential, you would see differences between these two experimental paradigms. And I'm just going to show you here what happens after depolarization in barium. Actually, almost no sparklet activity. Calcium is in the external solution. Barium was just used during the depolarization. But look what happens when the depolarization was produced in the presence of calcium. So this site is just going nuts. Calcium is coming in like crazy to this particular set of locations. So suggesting that once these two channels touch each other, and if that interaction persists, guys, these channels are going to open and close a lot, and a lot of calcium can go into these cells. Okay? So that interaction will be long-lasting. Now, if calcium is necessary, the first candidate you think is calmodulin or something that binds calcium is important in this process. And one thing that I would tell you is that yes, it is calmodulin, and the way we did the experiment was using two different approaches. First, we used calmodulin inhibitory peptide derived from MLCK, the myosin light chain kinase. We dialyzed the cells with about 10 micromolar of this peptide and ran the same protocol that you see above that triggered an increase in venous fluorescence, reconstitution of the channel, dimer coupling of the two adjacent channels. And if we did the same experiment now with cells that were expressing a dominant negative of calmodulin, calmodulin 1, 2, 3, 4, this is a calmodulin that has been mutated not to bind any calcium in any one of these 4 binding sites of calcium, we get exactly the same result. So what this tells us is that calmodulin is critical in this process. Now there are 2 types of pools of calmodulin. One is pool of calmodulin, as David Yu elegantly demonstrated, that is pre-associated with the channel, primarily the IQ domain. And another pool of calmodulin that is cytosolic or just near the channel, but is not necessarily associated with the channel. And what this experiment is telling you is that the calmodulin inhibitory peptide, which presumably is targeting the soluble calmodulin, and calmodulin 1, 2, 3, 4, which is targeting both the pre-associated and the cytosolic calmodulin, could be important in this coupling fusion problem. To be honest, we don't have an answer on how to molecularly how this is happening, but what we know is that it's not just one or the other. It seems like calmodulin could be coming from multiple sources.
So it's interesting that it's not reversible, because you would assume, with unbinding of calcium, it might reverse.
At least not in this experiment, but I'll show you some prep experiments in which it is reversible. But the interactions between the channels last longer than the calcium signal that evoked it. So there seems to be a little bit of memory in the system. This is a summary of the data for control conditions, calmodulin 1, 2, 3, 4, and MLCK, and the bar plot of that. Now there are 2 possible sites of two possible sites of interactions with calmodulin. One, as I mentioned early on, is the IQ domain, which has been shown by many investigators to be the site critically involved in the process of calcium-dependent activation, and the pre-IQ domain here in green that also can bind calcium and also is involved in calcium-dependent activation. So which one it is? So what we did was to selectively mutate, and we did lots of these experiments. We spent about 6 months to a year trying multiple mutants to try to figure out, which domain was responsible for these channel-to-channel interactions. Was it the IQ domain or the pre-IQ domain, or both? Here, the first set of experiments that I'll show you is a set of experiments with a triple mutant that has been shown by Georg Stresnik and also experiments by the U Lab to be involved in preventing at least the binding of calmodulin to the pre-IQ domain. Not touching what happens in terms of the pre-IQ domain then in terms of calmodulin. Well, what we found was the following, is that any mutation, in fact the other way around, the pre-IQ domain that was the one that was altered in when we modified the IQ calmodulin binding to the IQ domain, we got normal reconstitution of the channel as we depolarize the cell from -60, -20, 0, and +20 millivolts. Compare that to what happens under control conditions. This is the short form of the channel. And then when we did the triple mutant, the AAE mutant that was targeted only to the pre-IQ domain of the channel, we saw no change whatsoever in terms of split venous fluorescence. So what it tells us is that potentially not only the process of calcium-dependent inactivation, but also the dimerization of the channel could be controlled by slightly different mechanisms. In other words, the channel can undergo calcium-dependent activation normally, and at the same time, by a different entity, a structural entity, be involved in this process of dimerization. PIQ domain, remember that, be important. It turns out that it's the same situation with CAV 1.2 channels. This is the summary data from these experiments. Now, so far, all the data that I have shown you with regards to the venous reconstitution was done using the short form of the channel. It's right here. With its EF hand, VIQ domain, in green, in purple, the IQ domain. Now we wanted to take a look at what happens now with the long form. And more importantly, we wanted to make sure that we could look at the long form, again with the venous, the N venous and the C venous, but now with this distal C-tail regulatory domain that, as I told you, modulated the interactions between calmodulin and the IQ domain and the PIQ domain in the long form of the channel. What we found was, number one, is that with the long form of the channel, there was absolutely no reconstitution. None. Zero. And I will tell you something that I think was a lesson to me. It was very humbling. Keep in mind, we submitted a paper about 2 months ago, and it was brought up to our attention that we were using a clone that had one amino acid mutation. There was an error that was introduced by a cloning, the cloning procedure. Our gene did not mention this, so we had to go back and repeat all these experiments with not only data that we've done, that I show you, but now with the corrected one. And I'm happy to inform you that we got exactly the same result, but it was a valid experiment. Just one thing, don't trust necessarily what's in that gene. Do your homework and try to figure out whether there are other genes that are being posted that obviously had the mutations corrected. We thought the mutation was fine, our clone was fine, but it was not. So we repeated everything on that. But as I said, everything worked fine, so we're moving on. But one thing that we saw was that with the CAV 1.3 long form of the channel, the delta 116, we could completely reconstitute the dimerization of the channel, the CAV 1.3 long, doing membrane depolarization in the presence of calcium. Exactly the same features that the short form of the channel has. So what that tells us is that long tail serves as a steric, to prevent the channel interactions, presumably by blocking interactions between adjacent channels at the pre-IQ domain. One of the reasons that we're so believe so strongly the IQ domain, a pre-IQ domain is involved in this, the channel-to-channel interactions, not solely based on our data. Susan Hamilton and Fallon at Baylor University have very nice data, beautiful crystallographic data showing dimers of pre-IQ domain of CAV 1.2 channels that are cross-linked by calmodulin, and they did very nice measurements of these dimers. So there is precedent at the in vitro level, at the molecular level, at the atomic level, of dimers forming between CAV 1.2 channels. CAV 1.3 channels are slightly different, but this section of the channel is pretty much identical to what they were looking at. So we feel good about it. It's just to say, data summarizing our experiments with the long form of the channel. We know dimerization and CAV1 chopping off the last 116 amino acids, rescuing everything. Now, I told you early on that CAV 1.3 channels contribute to the action potential of SA node cells. So we took our human usable pluripotent stem cells, we infected them now with these channels that can form dimers after eliminating them with 488 light, and we try to see what happened to the action potential of these nodal light cells. We don't have the technology yet to isolate SA node cells from adult mice, but we'll get there. But at least this is a nice proof of concept in terms of what happened to the activity of these cells when you bring together CAV 1.3 channels. And here I'm showing you an action potential of these cells, the nodal cells. and the rate of depolarization is the first derivative of that action potential. And look what happens after elimination with 488. There is a subtle but significant increase in the duration of the action potential, and the rate of depolarization increased significantly too. So bringing together these channels has functional consequences in potential in nodal cells. We're going to be testing, and we're in fact started experiments trying to test this hypothesis in adult myocytes. And most importantly, testing the hypothesis that if we put back a CAV 1.3 channel that cannot form dimers, whether anything between these, anything that we think could bring these two channels together could also alter under physiological conditions pacemaking activity by SA node cells. The reason we think this could be relevant is because signal that we know controls pacemaking of the heart is activation of beta-adrenergic receptor during autonomic nervous system regulation of the heart. And when that happens, when cyclic AMP levels increase in SA node cells, what happens is that there's an increase in pacemaking activity. And look what happens with venous fluorescence when you add force cooling, which increases cyclic AMP level. Boom, venous reconstitution goes up. Now look what happens now in terms of CAV 1.2 channel venous fluorescence versus of isotope channel. This is the other channel that I told you was involved in the rapid depolarization phase of SA node cells. Fluorescence increases quite a bit. And through other experiments, I can tell you that we know that it's due to activation of protein kinase A and potentially phosphorylation of this channel. Now, exactly how phosphorylation of the channel is increasing the probability that these channels come together at a higher rate, we don't know, but we're doing some experiments to that extent to figure that out. Now, as I told you, we were interested to figure out, well, okay, so in nodal cells, CAV 1.3 channels, particularly the short form, can form dimers, can drag, can open and close cooperatively. What about neurons? So we took hippocampal neurons. First, we transfected them with CAV 1.3 short venous, and the N terminus and also the C terminus of venous, and tried to see whether under basal conditions, baseline, we can see reconstitution. And you see here in green is venous, and red is just plain RFP that is expressed to identify the cytosol of the cell. And look at pretty much everywhere that we look along the membrane of the cell, in the cell body, the axon, you see the dendrites, you see nice levels of venous reconstitution. Went back again, now in these cells, in the hippocampal neurons, and use our dimerization system. This is a neuron that is firing nicely and very happily action potentials. Hippocampal neurons before elimination with 488 nanometer light that would induce dimerization of the channels, and look what happens after we induce the dimerization of the channels. Cells goes nuts. One action potential after the other. One thing is that if you add nifedipine or any other dehydropyridine, the activity goes down, way down, in terms of action potential production. I want to end just by telling you that, yeah, CAV 1.2 channels coupled to now these channels play a prominent role not only in SA node cells, but also in ventricular myocytes. These channels clustered. The clusters are slightly bigger, about 6 to 10 channels along the Z lines, the T-tubules of ventricular myocytes. And this is a super resolution image of a ventricular myocyte that was labeled with an antibody against CAV 1.2. And it seems as based on single bleaching of EGFP tag channels that, and this is all done, by the way, in these freshly dissociated ventricular myocytes. What we did to tag these channels is that we took beta subunit, in this case we took beta 2, we fused it to EGFP, packed it into a veno-associated virus, injected the virus into the myocyte, dissociated the cells, and then did these experiments. And these are the clusters that we saw through the cell, monitor over time. And as I said, based on this type of analysis, we estimated that about 6 to 10 channels form our cluster along the C lines of the cells. And Yoram, this goes to your question right now. We know that calcium alone can induce the interaction. These are all FRET experiments between CAV1.2 EGFP and CAV1.3 tag. Nice increase in calcium is associated with an increase in FRET, and that interaction subsides as calcium goes down. So it's dynamic until you write calcium. We did a very detailed analysis of the relationship between the intracellular calcium concentration and the change in FRET with a V50 of around 254 nanomolar. So at rest, we saw very little level, at resting levels of calcium, 100 nanomolar, very little level of CAV1.2 to CAV1.2 channel interactions based on FRET fluorescence. But at a level of a spark, so when you see a spark is a release of calcium from a ryanodine receptor, produces a local increase in calcium that's been measured using flow around 254, you get about 50% of the maximum FRET response. So what that means is that if you have a very high rate of spark activity under control conditions during diastole, you can be priming a lot of the channels to touch each other, and probably, or potentially, when the next action potential comes, open at a higher level of open probability. Here's the most important thing, is that what we found was, this is something that is just very repeatable experiment by experiment, is that if we do an experiment in which we record the calcium current that's produced by the opening of these channels and the FRET signal that results from the interaction of these two channels, is that these interactions seem to last longer than the local calcium that creates them, rising with some delay after the onset of this the calcium current, but we're something at a slightly lower rate than with the current, so it may be that this is higher here. But the thing is that it descends then to a plateau that persists and then decays here as the cell is repolarized, with a potential tail here as the cell is repolarized to back to minus 80 millivolts. So there seems to be some memory in the system with regards to the duration of these improved channel interactions. So you can envision that as you're increasing pacing activity, more action potentials arrive as a function of time, that the higher you go, the probability of finding channels that are touching each other and that potentially have a higher open probability goes up. So this could be another factor contributing to rate-dependent increases in the calcium influx that are seen in cardiac myocytes. We're testing that hypothesis. Faction final, you want to tell you that like CAV1.2 short channels of the alpha, CAV1.2 channel coupling requires not only Cal modeling, but also the pre-IQ domain of the channel. It seems to be exactly the same model. So how do you think things are working here? So what we think is that channels under resting conditions are just happily, in the membrane doing their, in the closed state with some Cal modeling pre-associated, particularly to the IQ domain already there. Some cytosolic Cal modeling around in the vicinity of the channel. This says CAV1.2, but it applies to CAV1.3. As the membrane depolarizes, one channel opens that brings calcium into the cell. There's a local increase in calcium right here around the mouth of the channel. This incoming calcium binds to Cal modeling, and that somehow then leads to calcium can only bound to their PIQ to increase the probability of interactions between two adjacent channels in one of these clusters. And the channels will remain functionally coupled until, obviously, there's calcium-dependent inactivation of the channel, which I told you was untouched by this system. Happens, the channel is closed, calcium goes down, and as calcium goes down, these channels will tend to dissociate. However, this dissociation will take a little bit longer than the decreasing intracellular calcium. So these here, what we're going to call prime channel, or in the case that the nocturnal action potential shows up, part of that molecular memory that I showed potentially could be important in terms of what happens when during successive trains of action potential. If calcium remains low and for long enough, then the channels dissociate fully, and then the whole cycle begins once again. So how do we think things are happening? So here is just a closed model in terms of what happens to FRET channel interactions. At low stimulation rates, action potentials arrive into CAV1.2 to CAV1.2, CAV1.3, CAV1.3 interactions increase, that decay of time going fully dissociated channels, and so on and so forth. As the heart rate increases, then their action potentials start coming closer together when channels are still interacting with one another, particularly at very high stimulation rates, where then you can have a buildup of the number of channels that are interacting and therefore opening and potentially coupling out. I'll stop here and just thank the people that actually did the work. Here. And take questions. These are Cannon Beach here, Oregon. Not Seattle, but love it.
Not Davis.
No, David, that's for sure. Rosie Dixon did all the CAB 1.2 work. Claudia Moreno did all the CAB 1.3 work on hippocampal neurons and also in model cells. Jimena and Ken Yuan were two fantastic technicians that worked in my laboratory, generated all the constructs that we saw that we used in these experiments. And my dear colleague Mark Binder, neuroscientist, who enticed me to do some experiments in hippocampal neurons. And we're excited that we have done. Thank you.
So, if you have two, you show the mechanism for two to come together, but in your clusters you have 6 to 10. So what's bringing twos together?
Dean has asked me that question. It's a great question. Pretty much all the tools that I show you are built around dimers, right? Split Venus and also the dimerization system. But based on the electrophysiology and the sparkling data, I think that maybe more of that than one channel can touch each other. From a molecular point of view, I don't know yet whether what you have is a set of dimers or trimers, or you can have trimers or higher order oligomers interacting more or less on the same site. I have the feeling that it's going to be, in most cases, dimers of dimers. I may be biased by the split Venus and the cryptochrome data. But we've asked some structural biologists, Dan Miner and Susan Hamilton, to see if there are other binding sites that will support the view that you have multiple binding sites for the C-tails where you can have trimers, tetramers, etc., etc. Here's another possibility, is that you have accessory subunit, that protein or scaffolding protein that serves as the anchoring point where these channels touch each other and therefore increase their quality. I don't know to what extent, and then this is all related to the question of how one channel touching to the other leads to an increase in open quality. If you, as you're probably aware, the experiments with GABA and AMPA receptors, there is some pooling that is being thought as, binds glutamate, for example. The pocket just closes and that potentially leads to the opening of the gate. Another indication that some pooling could be involved in the triggering of two or multiple channels comes from what we know about STIM and ORI1. When you have STIM binding to ORI, and presumably it's just the pooling what opens it. That said, then the hypothesis that there is an intermediary protein that just simply is critical in touching itself without any pooling or any molecular motion involving the C-tails that are linked together could lead to the gating of the channel. I think is less likely. So I think that while it's possible that there is a molecule that brings the channels together, it is channel-to-channel interactions and some motions from one channel to the other that are translating to the opening of the secondary channel. And I think one of your students has the key to this. Robin Shaw found a protein, BEAM1, that clusters CAB 1.2 channels. And we've been collaborating with Robin, and we find that if you don't have BEAM1, CAB 1.2 channels are all over the place. You don't see the clusters that you see here, and you don't see the level of, so far, cooperativity that you see from this point. done beautiful work.
Well, Rolkin has been trying to generalize the concept of trafficking to calcium channel loss.
Exactly.
So that protein is in TSA cells?
It is.
Uh-huh.
D1 is there.
So you actually didn't say it, but if you put your tag on the N terminus of one and the C terminus of the other, do you ever
No.
see it?
Never.
And what about if you put the tag, if you mix one long and one short? Have you ever seen?
No, and that's an experiment that is a very good experiment because it would be very interesting to see if then just by having the short there, then it would open, just because it pulls or whatever. One thing that I can tell you is that, as you know, because I presented this in other places, that if you have a channel that opens with a higher open probability, you mutate the calcium channel so that it opens for a longer time and more frequently, and you fuse it to a wild type channel. The wild type channel starts behaving like the mutant channel. So the channel that opens with the highest probability, or that opens first, has it's easier to bring to the open state, seems to be the dominant channel. Not the other way around, at least in the context of CAV 1.2 channel. So you can amplify the signal by bringing channels together. It doesn't take that much. Yes.
So you mentioned the beta adrenergic receptor increases CA1.2 dimerization. Is that specific for that isoform or 1.3 as well?
1.3 as well. Not the CAV 1.3 long, but the short, yes. So far, we haven't found a way to bring CAV 1.3 long channels together and have them form functional dimers. No way around. And I was happy to see that, because to some extent, we started working with the short form and did the same thing as the CAV 1.2, and said, wait a minute, is everything that we're going to link together? And in the N terminus, that was one thing, but then when the long form didn't do it, that it was a lot of fun.
So what about, first of all, thanks. This is very useful. What about inactivation? Do these channels have to separate in order to inactivate?
No.
Or they inactivate faster than the memory time?
They will inactivate at normal times if the channels are associated together, are linked together.
And the other question, do you think that beta adrenergic effect is in a major way through this process, or through the increase in the single channel conductance?
I don't know. I don't know the extent of which this process is caused. I don't know how much of this dimerization contributes to the
Beta adrenergic
increase in the calcium.
It's an interesting question, because the other channel that is sensitive to calcium and under control of beta adrenergic stimulation is IKS. And, it's intriguing to think about IKS in the same kind of
Yeah, I don't know. That's a good.
So try IKS this.
Yeah. Should I would. I will. Yeah. Thank you guys.